Welcome. This tutorial will use plasmodb.org to describe the features of a gene record page. The quickest way to a gene record page is to enter the gene ID in the quick search box at the top of the page. You can also reach a gene record page by clicking on the ID in any result table. This is the gene record page for dihydrofolate reductase. At the top of the page is a header containing the gene ID, the gene product name, and quick links to adding user comments and adding the gene to your basket or favorites page. The box on the right offers links for downloading the gene information and expanding or collapsing information on the page. The gene record page has 5 sections, and you will need to scroll down to view all the sections. Overview, Annotation, Protein, Expression, and Sequence. Each section is a list of links to all the data available in the database. Data is either represented graphically or in tabular form. Click Show next to the description to expand and view the data. Click Data Sources on the right to see information about the origin of that data. The first option in the Overview section is a genomic context graphic. Dihydrofolate reductase is highlighted in yellow. Its location in the genome is shown in relation to the surrounding genes, with the scale above the graphic representing the genomic or chromosomal location. Below the DHFR graphic are several other tracks of data showing the same region for the other available strains. The gray lines between the tracks represent synteny across strains. Scroll down in the Genomic Context window to see more data tracks, such as the SNP data. The SNP data is also available in tabular form by clicking Show next to the data track title. To collapse the table, click Hide. Another useful feature in the Overview section is the multiple sequence alignment. This tool will produce an alignment for the specified nucleotide positions across the species that you choose. From the Annotation section, you can link to a form for adding comments on the gene page. Other useful tables include external links to other databases, Go terms, and names, previous identifiers, and aliases. The Protein section is below the Annotation section and contains several expandable links to useful tables and graphs. The first option is a graphic that is similar to the Genomic Context graphic but shows protein features. In the Protein Features graphic, data tracks display data such as proteomics peptides, interprodomains, and BLAST-P hits. Data tracks that do not contain data will appear blank, such as this transmembrane domain track for DHFR, which is not a membrane-bound protein. Proteomics data is also available in tabular form. The Expression section contains genomic scale experimental data concerning gene expression. Data is arranged by experiment. Clicking Show leads to graphs and charts of DHFR expression under the conditions of the experiment. Expand the first option, for example, to view the pattern of DHFR expression during the erythrocytic stage over a 50-hour time course. The Sequence section contains links for predicted protein and mRNA, as well as genomic and coding sequence. These links are good for a quick cut and paste. You can download sequences in FASTA format from the gene page header. Thank you for watching this tutorial. Please contact us with any questions or suggestions.